Welcome, Sleuths. I'm so excited to have you here. If you don't personally know who I am, my name is Heather Kenney. I'm a dental hygienist. I'm holistic certified, and I am the lead microbial investigator here at the Saliva Sleuths community. I started this community for the curious clinicians who want to learn about the oral microbiome and the oral systemic connection. I host monthly webinars, which you are attending. These are right now public, but eventually I'm hoping to just host them for my community to explore and investigate all things root cause and oral systemic disease related. I work in the oral microbiome space. I am the clinical trainer at Bristol Health, which is a salivary testing, and I do all of the onboarding for any practices who are nationwide and in Canada. I have people in Dominican Republic as well, and other countries that are starting to use salivary diagnostics. So what I found in doing that process is there's a huge gap of education around the oral microbiome, the oral systemic connection, and how to balance the ecosystem and going beyond just antibiotics. So I tend to be more holistic. So I try to specialize on trying to find ways that we need to pay attention to, like salivary flow and pH, and putting the right products in for the certain patient, dealing with airway and gut issues, and maybe bringing some polyphenols or essential oils or, probiotics and supplements into the picture to help us balance that ecosystem that is so impactful for oral and systemic health. I'm so excited tonight to be stepping through the looking glass and exploring the fascinating world of microbial curiosities with one of my favorite microbial investigators here at Saliva Sleuths, Holly Moons. Holly and I meet regularly. We're the regular goers at my open microbe nights, and sometimes it's just her and I, and it's a super fun conversation. I host open microbe nights bi-weekly, and we talk about research articles, case studies, different topics on oral systemic, and it's really an open conversation for everybody to kind of share and learn from each other. Holly has dedicated over 26 years in the field to periodontal therapy. She works in a perio office, which has also led her to be a really respectable voice in oral microbiology, research, and education. She served in multiple leadership roles on local and state dental hygiene boards. She participates in numerous study clubs, including Saliva Sleuths Community, and continues to publish and speak nationally on the emerging concepts of oral health. What truly sets Holly apart is her passion for understanding the microbial world beyond what we have traditionally learned in dentistry. Her work focuses on polymicrobial synergy, pathogenic virulence factors, and the complex interactions that shape the oral ecosystem and biofilm behavior, disease expression pathways as well. Many of you may have already read some of her recent publications. She just had one come out on Tannerella forsythia that was featured in January and in March in the RDH magazine. Check that out if you have not. It's well worth the read. As dental professionals in the space, our mission is to reduce oral systemic disease, yet we really weren't taught the deeper stories behind the pathogens in our perio classes. So trying to manage that every day like we do, I'm hoping to give you some great insights through this course. With advanced salivary diagnostics and oral microbiome testing that's actually more accessible, we're actually uncovering some really cool microbial clues that challenge more conventional thinking and raise new questions on how we educate and communicate with our patients. So tonight, Holly will take us down the rabbit hole to examine some of the oral microbiome's most intriguing and virulent organisms. We'll be exploring how these microbes survive, adapt, communicate, and contribute to disease, and understand how these mechanisms can help us become more competent clinicians and educators. So whether you're new to salivary diagnostics or already deep into the microbial investigations, this webinar will provide a practical insight fascinating science, and plenty of new talking points for you to bring to your patients and your practices. So grab your notebook, put on your detective hat, and join us on this venture through the looking glass to uncover the hidden stories behind the microbes. So please welcome my dear friend and partner in the microbial nerdiness, Holly Wins.
Wow, that was amazing. Thank you. I'm gonna share my screen here. Holy cow. Thank you.
Hey, well deserved.
All right, so can you guys see? Let's see. Here we go. Can you see the whole thing?
Yes,
Okay.
perfect.
Wow, holy cow. Well, thank you guys. I feel like I'm in like-minded company because I see so many of my nerdy hygiene peeps. So honestly, thank you. What a pleasure. So, Saliva Sleuths. I want to invite you guys to Saliva Sleuths because this has been so good for me. Heather built this community, and it's just a bunch of us talking about the oral microbiome. She's built a ton of training modules. She goes over how to salivary test and protocols to follow, care plans. Again, we meet, bi-monthly and talk about microbes. Heather brings in all kinds of articles, and we just nerd out over the changes. It's super fun. And then there's all these monthly seminars. So I'd love to invite you guys join us. We would love to have you. So I want to start by just a funny little side and say, you know, I want to celebrate saliva because this is kind of what we do all day is we are glorified suctioners to some. Uh, but honestly, we're way more than that. And saliva is honestly magical. And so I really do want to highlight all of the magical things that saliva can do for us. And right now, saliva, it's biological data. With saliva, we have the capability of literally diagnosing or identifying biomarkers or systemic diseases. So, for example, right now, we can check cortisol levels for metabolic syndromes like Cushing's and Addison's. We can check for C-reactive proteins for cardiovascular disease risks. We can check for autoimmune diseases like Sjogren's. So instead of someone having, a painful lip biopsy, they can do a salivary test. There's equipment now to test glucose for diabetes. And some of that same hardware they're actually using to identify breast cancer patients. They can identify the HER2. And also, there's a glycoprotein, it's the CA15-3, which tells oncologists if cancer is progressing or if treatment is working, all through saliva from some of that glucose testing equipment. And then, of course, we've got oral health and disease, what we do all day. And so I would say for me, my challenge was I had been doing salivary testing for 26 years. And I've gone through a couple companies, but I've never felt confident really going over the test, sharing that information with the patient. The doctor would, I mean, we started with, Petri dishes, Dr. Rams at Temple, we would paper point, we would send it in a little agar dish, and it would go, and he would T-streak it in the lab and test antibiotics against it. And he would give us a report back, and it would tell us, what the bacteria were, if they were susceptible or resistant. But on that list, I didn't quite ever feel confident enough to know what that meant. I knew that that meant bone loss and bleeding, but to do a deep dive on, what Tannerella forsythia is, I just didn't know that. I didn't remember it from school. I didn't take the time to learn it. So as salivary testing becomes more and more common, I just needed a way to find my ground, my feet. I just wanted to be able to have talking points for patients. I wanted to feel confident. And so for me tonight, this course was really about me trying to figure out these bacteria and get to know them. Who were the players in the game? And I needed to figure out the cellular anatomy, like go back to basics, microbiology, the interactions with the host, the relationships that bacteria have together themselves. And I wanted really simple ways to talk to my patient because a lot of times learning this is, as one of my friends will say, this is really cute, but how do you use it in the laboratory? So how do I translate this test to my patient? I'm a very visual person, so you'll see throughout this whole course, I need visual aids. It helps me to understand really heavy concepts. And my dad will always say, you have to tell a story. If you tell a story, then those in your chair, they're going to understand it. It can't be over their heads. So to me, all of this felt very Alice in Wonderland-esque. You know, Alice is through the looking glass and on her journey, and in the looking glass world, in her version of Wonderland, everything is upside down. Sideways, you know, what's up is down, left is right. She's constantly chasing this rabbit, which to me is the way that I feel constantly changing or chasing down research. I feel like I have changed. I feel like as a clinician now, as I've read more about the biome and bacteria and the way that it relates to disease, I feel like I look at my patient differently now than I did then. And so I hope on this journey, I'm able to share with you some of the weird and crazy things that I came across as I've been reading studies and trying to educate myself. So it's really, this is for me, but I hope to share something with you. And so I want to really start with the beginning for us, which was Sokransky's complexes. So Dr. Sigmund Sokransky, he was a microbiologist up at Forsyth. And in 1998, which I thought was so funny because Titanic came out the year before, so it does not really feel like that long ago, he figured out that there were these bacteria that form communities and they are responsible for the disease process. So we will always refer to this as Sokransky's complexes. And so he was really defining the community and figuring out who was hanging out with who in the subgingival pocket. And if you look at this, even though it's not the typical pyramid you might see, you can almost imagine if a tooth were over here on the side. You've got your early pioneer species and the way that it grows outward. So way out on the farther end, you've got basically the red complex. So it's kind of neat to look at it from a little schematic, thinking in terms of how the biofilm forms. And then now it's growing. So the concepts now, this is what we have in 2024. This is the G.F. Moore's complexes, named after some of the scientists that discovered it. And you can see how complex and dynamic bacteria and their classifications and the relationships that they have. You can see how it's evolved, almost like a kaleidoscope. And they've really figured out now who are the healthy guys. Healthy guys weren't really in that early conversation. So now we've got healthy guys. We've added implants and we've grown the red complex. So where it used to be 3, there's now 4. So I just think how neat it is that Alice here, you know, the eat me, drink me, you know, growing taller, getting smaller. Our concept of biofilm is changing. So, you know, I just would say, you know, what's in our future? If we had Sokransky's before and we have GF Moore's now, I can only imagine what lab technology is going to continue to give us. What are we going to discover together as hygienists or doctors or dentists as we work together and really identify partnerships in these salivary samples? So we see it even in microscopy. What will the future bring us as we all collaborate and work together? And so for me, that's really exciting. The human microbiome has really changed. And again, this is where it helps me to be visual. If I'm going to look at the microbiome as a snapshot, we're going to usually talk about the species. And my mistake was when I would get back a salivary test, let's say from Bristol, knowing there are 834 species right now, according to Forsyth, I thought on that salivary test, I was going to get back a read with all 834. And I laugh right now because it seems so silly to think that was going to be there, but that was my mistake. That's what I thought. The reality is our oral microbiomes, we really have about 200 or less. And so that's what we're going to call the core. So 80% of the microbiome is dominated by that typical core. And And then you have the variable. This is going to be dependent on our own personal fingerprint, our lifestyles, our genes, our physiological differences, maybe where we live, etc. So we're going to have a little bit of a difference, but now I know. And so I want to say today that it is important to understand single pathogens, to understand these single species and really vet them out so that we can understand what kind of tools they're using to fight harder. But the reality is, we really need to know how they're working together. There are relationships that are happening that are maybe more important than the one. And so this is George. That's what they nickname him. This is Dr. Chen, again, up at Forsyth. And here he says we really have to look at all of the species and what they're doing together to understand how bacteria interact with the human host in the oral environment. So powerful as a single player here. You have the king can, really make or break the game. But at the same time, the game is also part of the conversation. So this is how I look at the biofilm now. I look at it as polymicrobial. It is multiple bacteria working together in synergy or in dysbiosis. So this is the PSD, the synergy and dysbiosis model. So Heather, here's the butterfly effect. Alice in Wonderland with the caterpillar. Going back to my beginnings in perio, we really focused on brushing and flossing, rubber tipping, great, meticulous, excellent hygiene and home care. And that, for a lot of patients, held them in a state of health. It was very powerful. And that has not changed. That is still our number one goal, to get the patient to own their hygiene and really do great oral care. But then over time, you start to see we get this dysbiosis conversation where certain pathogens are present that if they overwhelm an environment, they're able to start to trigger disease. So that's where perio, I feel, has changed. And then the conversation continues to grow because now we have commensal bacteria. We have the entire oralome. We're talking about virus and uh, fungus and protozoa. So we've got multiple different mechanisms working in the mouth, not just bacteria. And we also have our lifestyle, the disease that we carry, our own variables. So our own microbiomes and our own oxidative stress, our lifestyles, our diet, our genetics. So all of these are playing a part in how the microbiome looks. And then even now I keep seeing more and more about calcium and vitamin D, and we've got, ions and metabolites shared, the food that bacteria are using. One bacteria's byproduct is another's fuel. And so it's really neat to see the conversation continue to grow. Bacteria, I will tell my patients, when bacteria wake up, their plan is not necessarily to trigger disease. The plan is ideally to survive, and then secondly, to thrive. And so to thrive, it's almost like being invited to a tea party. You kind of have to think to yourself, okay, if I'm going to go to a tea party, what am I going to wear? What I'm going to eat? Do I need to bring a dish? What is the weather going to be like? Who's going to be there? Do I even want to go? So you start to ask yourself those questions. And to survive as bacteria, it's going to be the same kind of questions. Where are you going to live? Do you need to bring a dish, nutrients? Or do you feel safe enough to go? Do you like the people who are going? And then we've got adherence and biofilm. Who's going to this event? And then secondly, if we're going to thrive as bacteria, we're going to need to focus on basically the battle. And the questions are still the same. What are you going to wear? Who's going to be there? What's the weather going to be like? And so to thrive, you have to feel safe. You have an army and you are moving men around. You need to find safety. You need to have some version of shelter. And that's going to include the ability to move and hide, so eluding from the host. And you also need to battle well. You need to be able to fight back. And so we're going to have to fight the host, and we're going to need a lot of weaponry to do that. And so in my mind, a lot of this, to Heather, it looks like a garden. To me, it is very Game of Thrones. So it's very medieval in its look. And so I call these virulence factors. So the weapons that these bacteria use to thrive, not just survive. And I think of, if you see these castles here, this is what I would imagine pathogens looking like, queens in their castle, and the gardens and hedgerows and armies out in front of them to protect the castle and those within. So tonight we're going to go down the rabbit hole, and I'm going to try to give you really tiny parts and pieces of a couple of these guys. If we have time, we can do more. Otherwise, we'll just, keep going a different day. But I'm going to try to keep this short. So these are going to be, it was hard to cut these slides down, you guys. There's so much more to this. But at least you'll have a couple takeaways to take back to the operatory. So you're going to see we've got the red complex, we've got Fusobacterium, and then we've got Aggregatibacter, so blue and red at the same time. So before we start, I really want you to think about this biofilm as a spatial orientation. Again, so Kransky's pyramid, the complex is turned on its side, tooth, surface, building outward. If you are on the outer rim of a biofilm, you are up against the host. And so that's what I really want you to think about is whoever is on that outer rim, you have to be the fittest. You have to be the strongest. You have to be the smartest. It is going to take a level of strategic engineering and brain power to survive on that outer rim. Talk about a very dangerous environment. So those are going to be the best and the brightest of the pathogens. So if we start, to me, it looks like this. You have our 4 queens sitting at this war table, planning and scheming. Now, there may be times they work together, and there may be times they are scheming against each other. They might have secret side alliances that we don't know about. Everyone's moving parts and pieces on the board. And it all comes down to, going back to Alice in Wonderland, finding this hypothesis, this red queen hypothesis, which is a real theory. And that it's that species have to constantly evolve to survive against their rival species, survival of the fittest. And so the quote, Lewis Carroll's quote is, "Now here you see, it takes all the running you can do to keep in the same place. If you want to get somewhere else, you must run at least twice as fast as that." So highlighting the genetic achievements in order to get ahead. And these red complex queens, they're capable of producing toxic metabolites. They are capable of breaking down protein, of fermenting amino acids in an anaerobic space, aka below the tissue in the pocket. They have outer membrane or sheath vesicles. Get ready for this one, guys. That's going to be fun to talk about in a minute. They mimic the host, mimic each other. They evade and invade. So let's start with our first one, AA. My husband calls all of my dental nerds dental nuggets. And so you will see a literal chicken nugget here, because I hope that it brings you a smile. I have a weird, dad pun sense of humor. But I need a nickname for these guys. I need it to be simple. And so I came up with this Aggregatibacter Actinomycetum comitans as the nice neighbor. It's a keystone pathogen capable of interacting with loads of other species. In biofilm models, they've used models where they might have 6 bacteria or 10, and every time they introduce AA to the equation, the biofilm doesn't really shift. It's almost like AA goes unnoticed, whereas others may move in and you'll have drastic side effects. So that's why I call him the nice neighbors. You can see here strep on the side, AA showing up with cookies. Here I am. So this was actually discovered in 1912. Again, another Titanic reference. So it's when Titanic went down. So it's been around a while. And the name, if you ever get lost in who these guys are and what they do, always look to the name. And so if we break it down, we've got aggregate, which means aggregates, they clump. We've got actinomyces, which means ray fungus, because you can see here in this little microscopic image these little strands, these little fimbriae. So it was once thought to be a fungus, and so it was called ray fungus. And then comitans, meaning may accompany. And so it actually accompanies other actinomyces species, which are gram positive. So again, if you ever get lost, you just break down the word. Weird stuff that I figured out when I was studying these guys is during like culturing and colonies, this is a campnophile. I thought, okay, so I don't know what a campnophile is, so I had to do some research, looked it up. This is a bacteria that does best when it's in carbon dioxide. So it likes a little bit of carbon dioxide in the mix, not just a complete anaerobic mix. And so we start to really understand or see these, what scientists will say, food webs, a metabolite that is shared. And I found that carbon dioxide is produced by these four here, Fusobacterium, Eubacterium, Bacteroides, etc. So as the carbon dioxide is produced, it's used by these campnophilic carbon dioxide loving pathogens. So you've got AA here, strep mutans, wildly enough, actually loves carbon dioxide. And what it does is it processes it to be able to actually increase the pH of the environment so it can survive all of the acids around it, which I thought was really amazing. And then you've got here E. corrodens, Eikenella corrodens. So one man's trash is another's treasure. As we continue, another working concept that really blew my mind was, again, these bacteria, they do not wake up and think, I'm a cavity, a cavity bacteria, I'm gonna cause a cavity, or I'm a pereobacteria, I'm gonna cause perio. The pH for pereobacteria tends to be more alkaline. And then the pH for cavity causing bacteria tend to be more acidic. Of course, that makes sense, right? But it's for survival. So going back to the whole theory of survival and surviving and thriving, pH for bacteria in these communities has everything to do with their ability to live in a niche. And I thought that was really amazing to have that kind of hit home. So I will tell my patients that if there is someone that has a high risk for cavities, I let them know it is a survival mechanism. It is not their intention to cause cavities. It's to live and thrive. These colonies, this is another mistake that I had made. You can see this star-shaped colony. I thought that was AA, but it's actually the colony itself. And so you can see it broken down here. This is AA, this little guy with all of this webbing, that's the actual bacteria. And so as it is grown in an agar dish over and over and over again, it actually loses those little fimbriae, and you can see the community, the colony smooth out. And so that's why there are two different versions, a textured and then a smooth. And the textured portion, they found that it actually stuck aggressively to the agar dishes, to the plates that they were testing on. So it was very tenacious. And you start to hear that tenacious word, and you think too in hygiene, tenacious colonies, tenacious calculus. So it's very sticky, and it's almost Spider-Man and sticky like. So again, really, really interesting stuff, I thought. This is what I want to kind of present to you. This is thank you, Tuet, because Tuet is the one that gave me this idea to try to present a lot of information in hopefully an organized way. And this, think of this like your index card to kind of look at it as a snapshot. So AA is a gram-negative bacteria. Let's go back to the beginning. What's a gram-negative bacteria? So we've got gram-positive and gram-negative. Gram-positive has this huge peptidoglycan. That is the mesh of the cell. And then there's one cell membrane. Gram-negative, on the other hand, a lot more complex. You have two membranes, two cellular membranes. In the middle, we have this peptidoglycan. I kind of think of it like a sandwich. You've got these two pieces of bread, and then you've got the peanut butter and jelly inside. And on the outer rim of the cell membrane, this is where you really start to see these, O-specific side chains. You've got the lipopolysaccharide, etc. So that's where bacteria pack all of their weapons. All of their virulence factors are shuttled from the inner cell membranes out to that outer cell membrane, and they hold on to them or launch them as they see fit. The peptoglycan specifically is a really neat mesh on the inside of those two membranes. And what it is it's scaffolding for the cell. It's a molecular sieve, so it allows nutrients to come in or out. And it also allows the bacteria to grow. And so what happens is it unzips this peptidoglycan and rebuilds the wall. And so it's a target for a lot of antibiotics, for example. So if you've got an antibiotic that's able to target this peptidoglycan, the generation of it, then you're able to start to take out these pathogens. So really fascinating, the anatomy of the cell itself. It's a coccobacillus. All right, so now we're getting to morphology. What's a coccobacillus? So again, we've got to look to the words. The words here help give us clues. So this coccus has the O in it. So coccus bacteria are little round guys, like the O. Bacillus here has the two L's. So I think of it as like long rods, just like the two L's. Vibrio, so you can see where I'm going with it. Vibrio is a V. And then spirochete, if you want to call it that, or a spirochete, tomato, is a spiral. So again, look to the words. It releases outer membrane vesicles. This blew my mind. What is an outer membrane vesicle? So another mistake I used to make was when I looked at PG, I always saw these little, I called them balls of fire, and I thought those were gingipanes. It turns out those are outer membrane vesicles. And so the cells are able to gather up the cell membrane, turn it into this little, like it says here, a little toxic package of cargo, and it launches it out into the oral environment. And they are so small. It reminds me so much of Austin Power, like the mini me. They're little teeny tiny replicas of the adult pathogen, the original cell. And because they're so small, they're able to go farther into tissue. They can slice and dice before the pathogen gets there. They communicate. If someone's on antibiotics, they kind of put on a show in front of antibiotics and distract like a decoy. And all of these red complex pathogens have outer membrane vesicles. The other interesting thing is that FN also has it. So FN, an orange species, also has outer membrane vesicles. There may be more. That's just what I was able to learn about thus far. Really neat. Fimbriae. So fimbriae present. So AA, remember those pictures of those long fimbriae. So what is fimbriae? What is this type 4 pili system? So if we look at appendages on the outer cell membrane, we've got different ways for bacteria to stick and cling. So pili are these really long extensions, fibers, and it allows them to stick to heart vessels, land in the gut. It is what is used for bacterial conjugation. So that's what I call bacterial sexy time. And then it's also used to move around. And so it's capable of almost like a harpoon, throwing it out and then pulling. Maybe you could think of like the Addams family, like the hand as it's trying to, move across the table, and as it runs into certain stimuli, it can sense and touch, if it's coming into contact with another bacteria or a host cell. So it uses it to feel. And I loved this quote. Type 4 pili, it's recognized as one of the most versatile and powerful molecular machines in the microbial world. Thought that was so powerful a statement. 4 gram negative only. Fimbriae, on the other hand, are more like Velcro. And so these are like the little bristles. And so they're capable of helping to build the biofilm. Everyone's kind of sticking together Velcro style. And then you have flagella, which are long strands. You might have one, you might have bundles of flagella, and it allows the bacteria to move. So bacteria use these appendages to really stick and form those biofilms. Here we can see that AA has an arsenal of different appendages, and I love a visual. Again, love the way that this stuff looks. You can see jutting out from the cell membrane all of these little strands capable of binding to host cells and teeth, resisting that mechanical removal. Again, going back to that tenacious. So we hear a lot about AA and PG being SRP resistant, not just because they're in the bloodstream, but because they are really tenacious. And that's what they'll use again to interact with the host. So AA is also saccharolytic and proteolytic. So what does this mean? So as this AA being a nice neighbor, AA, I would also tell patients, is it's very dual in its effectiveness. It's a little bit of this and a little bit of that. And so what I mean is it's an early colonizer. It will form along, again, nice neighbor, along those strep species next to the tooth. So these pathogens, even though it's in a red complex, it's not always going to be subgingival, which kind of blows my mind. So we can put these in categories, but you do have to allow yourself the freedom to go. There's a little wiggle room. So to be saccharolytic means you're going to eat sugar. Sugar's your preference. And so here in this little picture, you can see specific early pioneer bacteria releasing lactic acid, feeding this AA, reducing other species like Veillonella. So just the act of AA being present can change the biofilm. Now, if you've got bacteria that are, here we've got high levels of lactate, you can see how we've got a shift from one to the other. So if AA is gone, then we have Veillonella take over. And so we've got this, fuel source. Being proteolytic means you're going to be breaking down proteins. And so this is where we really start to say to the patient, these are the bacteria that cause the bleeding and the bone loss. And I would also add, another little key is I will tell patients the same bacteria that cause bleeding and bone loss are the same bacteria that cause dementia, Alzheimer's, heart attack, stroke, cancer, etc. So it's easy to kind of in like one sentence give them, this is the stuff that's causing disease, not just oral disease, but chronic systemic disease too. So here it is eating protein. This is its fuel choice as it's a late colonizer. So early colonizing and late colonizing. Loves sugar, loves protein. So why is it going after protein? Well, the really important reason is to get the iron out of red blood cells. So when you see bleeding, these bacteria are using enzymes, proteases, collagenases, anything ending in ase is an enzyme. And they're using these enzymes to break the host tissue down, the gum tissue, etc. And they're just going for the heme. So they want the blood. They want to go after that iron. So for AA being able to do both of these, it allows for it to be metabolically more flexible. You've got a broader range of nutrient sources. And what's interesting is you think about what is in the sulcus versus what is in the oral environment outside of the pocket. Well, if someone's eating food, you're going to be encountering more sugars. So those bacteria that are above the gum line are going to be more of a saccharolytic type bacteria. Whereas if you're in the pocket, you're not going to have access to those sugars. You're going to need to use protein as your next fuel source. So here, competitive advantage. Talk about being virulent when you've got the ability to do all of these things. So here's my image of what I would imagine these pathogens in a pocket doing is you don't ever want to overwhelm the immune system and ignite the immune system to call the fire alarm, for example, because if the immune system's activated, it's designed to hunt and kill, and then you have this battle. No, 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 no. Pathogens, they just want to sit around this campfire and warm their little toes and have s'mores and share stories. So they love a nice, slow roast. So that's how I look at pathogens. AA is also nitrate reducing. So not all healthy bacteria, they're not the only ones that are breaking down nitrate. We've got other bacteria that are breaking down nitrate as well. And bacteria don't necessarily breathe, but they use oxygen in a different way. So AA is a facultative anaerobe and microaerophilic. Again, did not quite know what that meant. So we breathe air. It's what we need to live our lives. If we're not breathing air, we are not alive. And so by breathing air, most of it's nitrogen, some of it's oxygen, and it is our energy source. Bacteria, on the other hand, they don't have little gills, but they have electrons. And chemically, electrons become a currency for the ability to make energy. And so a lot of times the end terminal of this electron transport means you might have an electron end at oxygen or sulfur or nitrate. And again, what it means is energy. And so if we go down the line, this obligate aerobe, to be an aerobe is bacteria that need oxygen. Now, if you are breathing oxygen as a bacteria, you're using it, this is going to be an energy source that's really easy to get a hold of. And when you do get it, it's got a lot of fuel behind it. So they're capable of creating more energy with that oxygen. You've got microaerophilic, which is just using a little bit of oxygen. You're just now starting to dip your toe down in that water where there's none. You've got facultative anaerobes, which use everything. They might use oxygen, they might not use oxygen. And then you have an obligate anaerobe, bacteria that cannot survive in the presence of oxygen. And so we've got AA here with being a capnophile, and we learned earlier it likes a little bit of that carbon dioxide. I think of it as, nutmeg. Someone's cooking there. If you have something wonderful, and you might say, what is that spice? Why is this so good? Well, it's a little bit of nutmeg that's in there. To me, carbon dioxide is that nutmeg for AA. And so you can see here oxygen creating way more energy. So the ability for a bacteria to be a facultative anaerobe gives it that ability to be more dynamic, to be in different parts of the mouth at the same time. And these bacteria specifically, they use their own versions of enzymes to neutralize oxygen. So some of them are able to live above the gum line very safely. And they can also survive these oxidative bursts by white blood cells, so become way more persistent. So oxygen is the chemical warfare. You think about water picks, things, hydrogen peroxide type products, things that create oxygen. And now it can be either the thing that a bacteria breathes, or it can be the toxic mechanism in which they die. AA has been with us for a long time. We can get it when we're infants. It grows with us. And now we know its arrival in the biofilm is early and late. It's used to be known as just that classic first molar pathogen, but now we know it's involved in way more processes. And so you can see here, it's capable of living above and below the gum line. Again, guys, I'm so sorry I cut this short. So there's a little bit more that we might be able to do in the Saliva Sleuths to deep dive. But to summarize, AA has about 7 different serotypes. And here in the States, we have serotype B. This is the most virulent strain out of all of them, and it has this JP2, it's the genotype. So it's a very toxic virulence factor that this bacteria uses, and it's going to be more common in those that are under 35. I don't really like long lists like this of systemic diseases. This is kind of hard to see and for my brain to interpret and take in and remember. But I do want to put it out there in case any of you want to see a systemic disease that you're maybe really excited to learn about right now, so you can kind of look at these lists and see if one of these clicks. I just wanted to write down what I saw as I ran into research. So this is not AI. This was me pulling any kind of study that I could and putting it down. It might not match what classic literature says right now. It's just, again, just what I found in literature. And two that stood out: rheumatoid arthritis. We will have to come back to that a different day. Huge in AA, and I feel like autoimmune diseases are really reaching their pinnacle right now. I keep hearing about it more and more in a great way, highlighting it. And then we also have bacterial endocarditis, this HASIC group. And so there are groupings that are starting to stand out. And the A in HASIC is for AA, and these are bacteria that are linked with bacterial endocarditis. And most bacterial endocarditis infections are going to come from staph or strep. But this is a small little group of others that do also contribute to it. So again, maybe another microbe type. AA has a very well-known leukotoxin. I consider this the knight that is protecting the queen, and there is a ginormous broadsword that AA is going to use to protect the queen. And this leukotoxin is a surface secreted exotoxin. So it's on the outside of the cell membrane. It's used for close combat. So you can imagine it's stuck to the membrane there to protect the queen. This leukotoxin is really devastating to white blood cells specifically. So it's going to target and kill leukocytes, your neutrophils, lymphocytes, monocytes, the good army. The guys are part of our immune system that's designed to fight, and it has a very great way at wiping them out. It also can bind to antigens on these white blood cells, and it will poke holes in the white blood cells. How he does it, kind of just stabbing at that membrane. So as pathogens and that community support goes up, we see white blood cells and the immune support go down. There's not much involved with red blood cells, but it can cause pores in red blood cells, leading to hemolysis. And so it will definitely be a little part of this too. So it's not just white blood cells. AA. It has another toxic virulence factor, which is dispersin B. And dispersin B is the way in which it can actually escape a biofilm. So here you see off to the far left, AA sitting here above the gum line with strep. So again, the nice neighbors. And they might be hanging out for a long time, and you've got strep, happily producing that lactic acid, and AA is loving all of that sugar and attention, and AA is growing. Well, there might come a day where AA has kind of had its day. Strep is just thinking, I am so tired of AA, I just need a break. And so strep is going to produce hydrogen peroxide. And AA is going to think, you know what, I'm going to take my ball and I'm going home. I don't need this drama. And so AA will eventually move down into the subgingival terrain to try to escape the hydrogen peroxide. So here we have this dispersin B as it's an enzyme that is used to rupture that biofilm and allow it to migrate above or below. So you can see it's also off to the side. We've got that leukotoxin, which is upregulated when it starts to head south. And so AA also has these enzymes encoded by genes, again, used to degrade hydrogen peroxide. So many of our products create hydrogen peroxide. So you can see here if it has an enzyme that is coded to degrade hydrogen peroxide, we can pretty much assume that this is going to be, again, that very tenacious, tough to treat type of bacteria. And it will also protect PG in these microenvironments. So it has that protective mechanism. So moving on to the next pathogen. This is going to be the Heather and I always joke about, PG kind of having its day. So it's definitely the influencer bacteria. Here it is the master manipulator. And I always loved the movie Beetlejuice. It's showtime. So this is definitely the one that has been the most studied. Why? Because it's the easiest to study in the lab. So 10 to 1, 11 to 1 is more studied than TD or TF. So it's just had a lot of limelight shown on this bacteria. It is aggressive and it is very dangerous. We just happen to know a bunch more about it. So it is the causative agent for PD. I am surprised at how many saliva tests I have seen it or not. And it is not the only bacteria that's going to cause disease. The origin. So again, breaking it down into the words. Porphyria ramonis means purple unit. Purple unit, we'll get to that in a second. It's a gram-negative coccobacillus, so now we know round and rod together, releases those outer membrane vesicles, just like a classic red complex bacteria would. Gingipanes. So here we go with the gingipanes now. Some of them have capsules. Okay, so I'm going to stop for a second. Well, what's a capsule? So a capsule is mostly gram-negative anaerobic rod bacteria have these capsules, so PG and P intermedia. It's capsules there to support survival, and it is protection against the white blood cells, our neutrophils and our macrophages, and allows those particular species to have a higher virulence. So this is one more extra protective mechanism to survive on that outer rim. It's got fimbriae, so long attachments to stick to things. Proteolytic, all day long, loves that protein. It is not mobile. It's kind of stuck where it is. It can brainwash and control, but it's pretty much static where it is. It does not reduce nitrate, but it does reduce nitrite. Okay, so this is another weird scenario where thinking in terms of commensal bacteria that break down nitrate and nitrite. Well, this one is actually PG. The addition of nitrate can take it out, can harm it, can hurt it, can change the biofilm. But it's got these nitrite reducing reductases. It can use nitrite. So very complex in its way to break up that conversation. And it's an obligate anaerobe. It must be below the tissue. It does not want oxygen. So here are those purple units, purple unit in the colony dish, the Petri dish. So it's named after what it looked like. Loves a very nice alkaline environment. Again, talking about pH earlier and how periobacteria tend to like that alkalinity. It loves that iron, loves the red blood cells. And now we have a new nutrient, vitamin K. So I thought it was interesting because vitamin K is a growth factor for PG. But here you can see the bifidobacterium, a healthy species, actually consumes vitamin K. And so the healthier species that you've got going on are able to take away some of the growth factors for PG, for example. It is highly transmissible. AA can be passed on. PG can be passed on. What does that mean? It means when you're doing SRPs, a great post-op set of instructions is tonight is the night to not go home and have a make-out session. Tonight is the night you do not want to share your meal with kids and pass forks and spoons, etc. This is transmissible. Not a huge chlorhexidine fan. We have it in one of the offices, and I'm in the habit now of saying, hey, would you like to do, a 30-second swoosh with chlorhexidine, because I just want to try to get all of that planktonic bacteria that I just blew out of there. So I'm a little bit more sensitive to that now. It's a late colonizer and requires other bacteria to reduce that oxygen environment and anaerobe living below the tissue. PG, again, another tough slide for me because this is a lot of words. but I want to skim over this because it is very dangerous. So it's triggered by these polymicrobial communities. It will disturb the balance of the biofilm, brainwashing and controlling how things work and operate. It will degrade white blood cells, neutrophils. It escapes the host's ability to trap them and kill them. It produces hydrogen sulfide and methyl mercaptan. These are the two chemicals that lead to halitosis. It's capable of attaching to cells, invading in cells, and it can also stimulate and suppress the immune system. Again, this is that slow roast around the campfire. So it's great at stimulating enough trauma and inflammation to get the iron, to get the blood that it wants. It loves the nice swollen papilla, but it wants to keep that under wraps. It does not want to wake up that immune system. So it's a non-productive inflammation. Here's our systemic list here. Again, big dogs. I guess the biggest one is oral cancer to 36% if PG is present. Diabetes, rheumatoid arthritis, heart disease, of course, neurodegenerative disorders. So here's PG's appendages. Again, seeing these visuals, we've got PG here in the center, these little round guys, and all of these little appendages. This long guy right here, this is FN. And so we have these two kind of velcroing and sticking together and sharing metabolites. And hidden within this biofilm, all of these little V's, these are the outer membrane vesicles. And so now you kind of have like an up close visual of all of those vesicles in action. And these appendages, again, tenacious, they allow for attachment to host cells. And so what happens is the host cell, like a key, they are able to open up the host cell and climb on in. So here's PG in a cardiac cell. You can see all these little PGs on the outside of the cell. They want in. And so once they are in, the bacteria is enclosed in this phagosome, this sac-like component. But once it's there, it's cut off from its fuel source. So what's it going to do? It's not going to live long without resources. And you have, of course, this impending lysosome designed to come and fight it and kill it off. So what's it going to do? Well, it takes over and it decides, I'm going to induce cellular recycling because I'm going to survive if the cell's in this recycle regeneration type process. So what happens is the membrane, this autophagy, this membrane forms around PG. And now it's capable of literally with gingipanes, it's capable of breaking down that inner lining and using it as a fuel source. So it can live in that little sac on its own, feeding off the nutrients. So very parasitic in its nature. Favorite slide ever. So now we have the outer membrane vesicles, those little balls of fire like we saw earlier. And we also have gingipanes. So I'm a redhead, so I am a ginger. And so here we've got the Mad Hatter as a ginger, but all of these little bits and pieces, parts of the cargo that are in, now we have tiny Alice. So we've got these little tiny balls, these outer membrane vesicles, these teeny tiny mini replicas. So just like AA, we've got the same concept with it being the cutting, traveling farther, deeper into tissue. But now it's able to, with PG, with these gingipanes, it can stalk the outer membrane vesicle with the gingipanes, these enzymes. It can get rid of waste. So here you've got, this incredible picture of PG with all those balls of fire, all those outer membrane vesicles, and even a smaller version of the vesicles on the outside of the cell, literally climbing into another host cell, nuking tissue as it moves, using those to kind of light the fire and burn up the tissue so that it can migrate inward. PG, the gingipanes themselves, fascinating. So again, love enzymes. It is a cysteine protease toxin that it uses. Enzymes use to shred. These are their shredders. And you've got different versions of gingipane. You've got this KGP and you've got this RGP A and B. They are gingipanes that have affinities towards residues, the lysine and arginine residue. You do not have to know this, but at least you understand that there are multiple gingipanes and they're going for specific targets to help them to identify what they need to slice and dice. So they're located on the cell membrane, and these are very sensitive to oxygen. So again, PG's got to be in a no oxygen environment. So these gingipanes don't work well if there's a lot of oxygen. Responsible for most of its ability to break through protein and tissue. And these can actually degrade the white blood cells, your cytokines, your complement system, antioxidants. It loves antibodies as a fuel source. So you can see just in this photo, we've got these gingipanes producing in the vascular system this capillary permeability, inhibiting blood clots and increasing bleeding. This big juicy capillary. I just love it. Look how huge it is. So you can imagine PG in those tissues right now and creating this like slow inflammatory roast. So it's got all this access to red blood cells. And of course, it's going to prevent a blood clot. That's the last thing it wants. It wants blood flowing. So you can see in that photo how you can imagine what it would be doing. Next door, on the other hand, we've got recession. So lack of collagen, lack of tissue. We have the way in which it's going to cleave gum tissue collagen to get amino acids. So if PG were present, maybe this is what's adding to all of that recession that we see, degrading everything in sight, blood vessels and tissue. And then we have one more protease type toxin. We have sialidases. I'm going to come back to that guy later, but just remember PG has sialidase. One last thing about PG with the gingipanes are capable of, again, just like strep can produce hydrogen peroxide to get rid of AA, if AA, almost like new money, if AA shows up, PG is definitely old money. PG has been around. PG is a Rockefeller. And so if PG decides, I am so tired of AA, it's going to take the gingipanes and help debond AA off, and it changes the biofilm. Next target, Treponema denticola. These are the two. We've got TF right after this. Less known, less studied. I was fascinated to learn more about it. Heather calls this the swimmy guys, and I will never forget that. So to me, these guys are the movers and the shakers. These are the ones that are capable of moving the entire red complex around. It means treponema means turning thread. So in my mind, he's here spinning the yarn, moving around, related to diseases like Lyme disease and syphilis. So TD is a gram-negative, of course. It's helically coiled. So we've got a spirochete, so like a corkscrew. It does not have an outer membrane. Instead, it has a sheath. And so we're going to call these outer sheath vesicles. It does not have anything exposed on the outside as far as pili or appendages. Instead, it's internal. So there's an internal flagella. It's proteolytic. It wants tissue. It's highly motable. It can move, and it does not reduce nitrate. It is an obligate anaerobe. It must be below the tissue. It is as a spirochete, we do not know how many spirochetes there are. We have 10 right now. They suspect there are bunches. So I have a feeling that in the future, again, like GF Moore's complexes and the thought of biofilm as we see it growing, we will learn very quickly about more TD, more spirochetes that are causing perio. Very hard to culture, mostly found in periodontal disease. It loves hard surfaces like orthobrackets, lives below the tissue. And now we're going to start to see the way that TD layers. TD is on the outer rim of the entire biofilm. It is within the pool of saliva. So it is on the very, very outside. And so there are some concepts or classes I've taken where if you do SRP, that TD should fold, that you should see those numbers drop. And it makes sense if it's on the outside, you're getting it first with your desiccation therapy or your lasers. It's the guy on the outer rim. I'm going to go through this a little bit faster. What does it cause in the mouth? Of course, all kinds of abscesses, all kinds. But the complex that was interesting is it's part of the fusospiraketal complex. And so those are the three that are well known in eggnog. So you've got TD, FN, and PI. And I thought that was kind of neat to learn about. List of systemic diseases. And then now let's go into the enzymes, one of my favorite thing. So TD also has a cysteine protease. So we had PG with gingipanes. Well, guess what? TD has dentopanes. What's interesting is it uses it as long range. So I have this little arrow here. So it's capable of sending these enzymes far out into the environment. It's not quite attached to the cell membrane. So it's a fantastic weapon in that it can degrade the immune system and tissues. But TD has a better protease. It's a serine protease. And this is the big dog in TD. And it really is anchored to the outer membrane. It's used for that close combat, like the L-Tux in AA earlier, that broadsword. It's capable of close combat and is stable in oxygen. Very sticky and increases binding. And then you've got, of course, a list of the way that it's breaking down collagen and fibrin, thinking of that photo with the recession. It also has a sialidase. So now we're 2 for 3 as far as sialidases go. The internal flagella. So again, nothing on the outside to get it to move. How does this sucker move? The flagella are on the inside. Why would a pathogen put flagella on the inside? The reason is so that it is not attacked, so that it is not at risk of being targeted. And so it's got these little motors inside it internally to create these wave-like shapes. And so it can actually bore. And so we've got down below the gram-negative membrane. And now we know there's an inner and outer membrane. And here we have right in the center of it, your peptidoglycan. Remember the sandwich earlier? Well, now we've got it. Maybe this is the chip layer. And there's the one more extra ingredient that's going to go in that sandwich. So it allows it to propel forward. How it looks, I always think of like this little corkscrew. It bores. It's capable of boring into tissues. We learned earlier about syphilis, and syphilis is, the treponeme species. It's going to attack the brain. And so, of course, these little guys are going to bore into the brain. They're going to bore into tissue. And it uses chemotaxis. And so it can swim better when things are thicker. When the fluid is thicker, then it's got a little bit more resistance to kind of wiggle and crawl around. And so you can see in this microscopy photo all the little spirochetes in the middle. And again, I just hear Heather's voice. These are the swimmy guys. So they're on the outer edge of the biofilm in that pool, floating around on the move. Best picture of all time ever. Here is the red complex. So you can kind of see now, we don't even need any descriptors, no words necessary. You can look at these bacteria now and identify who they are. We've got Treponema denticola, the spirochetes. We've got these little coccobacillus, the Porphyromonas gingivalis guys. We have all of these little dots, the fireballs, the outer membrane and outer sheath vesicles. And you can even see TD here on the outer edge, almost like a coating. And then underneath it, we've got all of this PG. So who's this guy? Right here. The rot. Who's that guy? Underneath it all. On to the last of the red complex. And we have to go back to 1979, which is when Grease came out, for any of you that are old enough to remember the movie, Walkman, etc. And in 1979, this woman, she is my superhero. This is Dr. Ann Tanner, and she too was working at Forsyth at the time. and she found a bacteria that was unlike any bacteria she had ever found before, and it was found in perio abscesses. It would not grow in a monoculture, meaning on its own. It had to be co-streaked with FN. And so, as we discover now, she found Tannarella forsythia, named after her, Dr. Tanner, aka Tannarella, and forsythia as in forsyth. And this is the quietest, least studied of the red complex, again, because of how difficult it is to culture and study. So it took me about 2 years to really read everything I could about it and break it down. And it was really hard. And I had to read studies over and over again to really understand it. And then finally, things started to click. And so, TF, I want to share the mystery that is this wild, hidden, mysterious pathogen. And I always think of a dog underneath the Thanksgiving table looking for scraps. So TF is the scavenger. It must go out into the environment and hunt. So it is a taxonomic enigma. It does so many weird, different things. Dr. Tanner, just a side note, you can see here, she also found Campylobacter reptus. She found Campylobacter gracilis. She found Scardovia wigsei, which is a bacteria well known for childhood caries, which she's very proud of. So again, limited research. There was a time too when the strain that they studied got accidentally misplaced into another strains category. And so there was a time, there was about 10 years where all of the Tannarella forsythia studies that came out was inaccurately based off of the wrong strain. And so a lot of the knowledge and the research we have now is we're still getting caught up because we had this delay in that mix up. So it's a gram negative. It is rod shaped and sometimes fusiform. So it's capable of, depending on the environment, changing its shape and morphing a little bit. It has an S layer. So now we have a different layer. We had a capsule with PG, and we've got outer membrane vesicles, of course, no appendages. It is proteolytic and question mark. We'll come back to that. It is not mobile. TD is the mobile one. It does not reduce nitrate, and it is an obligate anaerobe. It is not wanting oxygen. So TF, you saw in that mass pile of bacteria, the biofilm. TF, every time I read about it, it's really interesting because without TF, you will not have the red complex. It is TF hangs out with FN, the orange species right below it. They love to share together. So you have this biofilm bestie scenario happening. And on top of TF, then you're capable of having PG and then TD. So I thought even within the red complex, those red queens, there is a hierarchy. And so without TF, you will not have that red complex. I thought, it's very foundational. Here's a neat little graph to show for myself, just to see the capability of what TF does in an environment. So you can see the numbers, the PG, TD, and TF on their own. TF, of course, you're going to have it be more prolific. And then you start to see over to the side as they form that polymicrobial mass, that relationship together, all of them at that war table, how aggressive and outrageous the numbers can get when they're working together as a team. TF, I keep hearing over and over again that if you find TF in a study, on a saliva test, that it has a lot to do with airway. And you're supposed to track airway. And for all of my airway friends here, I don't know if there's something that someone else knows about that I don't, but in all the time I was researching TF, I had the hardest time finding anything specific about airway. It just did not stand out anywhere. So for me, unless I read it in a study, unless I see it happen over and over again, it's not true. And so I put here that there is maybe an airway myth. And the reason is because there are others that are in the airway. It's not just TF. And so in this photo, you can see at the back of the throat here, we've got AA, we've got Fusobacterium, we've got PG, we've got strep, we've got Tannerella. And Heather may be able to highlight this in a different microbe night. You know, there's more than just TF at the back of the throat. So is TF the only airway guy? I say no, like probably the least out of all of them. TF also has, here we go again, patterns, cysteine proteases, serine proteases, and now let's finally talk about it, this sialidase. So now we're 3 for 3. This is a NAN-H. This is an enzyme that TF uses beautifully to break apart. This is how TF looks to me. You have a citadel, which is this fortress, and in the center, you've got the nobles, right? Medieval, times, so to speak. And you have to protect the nobles at all costs. And so you build outward with all of these walls. And on the outer rim, you have the peasants and the farmers and people coming in and out and traveling. So TF is built a lot like that. And then it has all of those layers, the same Na gram-negative wood. You've got inner and outer membranes, the same peptidoglycan, but now it has that S layer. So remember, guys, that peptidoglycan, look at these two colors that make up the peptidoglycan. We have these sugar molecules. They're called Murnac and Glicnac. They're technically called N-acetylmuramic acid, etc. We're going to abbreviate them. So we've got these sugar molecules bonded to peptides, which make up that mesh. Very important to remember as we move through these slides. The S layer is this right here. It looks like carbon fiber. And so I wanted to put in two versions of this S layer so you could kind of see the pattern shape. It's almost hard to look at, almost like checkerboard-esque. And that S layer, the way that it's built is it's a whole bunch of glycoproteins. Sugars, glyco, proteins, proteins. So over here on the far left, the visual is you've got the glycan and the protein. You might have in other bacteria glycolipids. And so these little guys connect to the outer membrane and create this shell. For archaea, they only have an S layer. There are no additional membranes. But for these gram-negative bacteria, you can see how we've got these glycoproteins attached to that outer membrane. And you've got different lattice makeups. So you've got squares, you've got hexagonals, you've got all these shapes that these glycoproteins can come together. Now, the neat thing about TF is it has a square-shaped lattice. And I wanted to put in, again, more microscopic visuals to kind of hit it home. You've got the wild type strain, which is the natural TF in its environment, and you can make out this very distinct kind of like textured, zippered, zigzagged kind of outer membrane versus the mutant without those genes making that. You can see how it blurs. And so we lose its ability to have that strong S layer, which is one of its virulent factors, protection in the biofilm. So again, we've got the intact, here's that zipper S layer versus mutant as it's blurring. So if we don't have an S layer intact, this is going to put that bacteria at risk. What makes TF even weirder? And again, guys, this took me like 2 years to figure out. So I finally figured it out. So here's that square lattice structure from the top down. TF is weird because it actually has two glycoproteins zippered together, making this crystal mesh. That's highly unusual to have two together. Very, very rare. So they call these TFSA and B, these two glycoproteins zippered together, tightly woven protection. One more weird thing, as if that wasn't enough, in microbiology, it's glycosylated. This is even rare. And so you can see this forest of all of these little parts and pieces, squares and triangles, and you kind of wonder, what in the heck are these? They're sugars. These guys are sugars, and it's what makes it a biofilm bestie with FN. FN needs those sugars, and TF, you'll find out in a minute what TF needs, which builds that relationship. This says everything. This is Dr. Carolyn Bertozzi. She is an amazing scientist. She is a glycobiologist. She is a chemist. She is a Nobel Prize winner. And it was her YouTube video describing and detailing out what glycoproteins are. Watching that is what made it all make sense. And so what she describes is our human cells have all kinds of little sugars on the outside of the cells. So you can see here we've got these glycolipids, glycoproteins, and then here's this forest of sugars. And so she loves and she's delighted by studying all of these sugars because we've got the ability to link things with cancer, etc. So she's really studying that. And we've got enzymes that can break those sugars down. And one of these sugars, this little purple diamond, it's just called sialic acid or SIAs. And they're kind of like the outer sugar on the outer rim of all of these glycoproteins. So TF, now that you can kind of see it as a visual, TF has the ability with these sialidases to break down our host sugars. So I say TF, it is proteolytic, yes. And we put question mark, question mark. And it's because it can also break down sugars, maybe not saccharidically like typical glycose, but it does break down our human sugars. And so this little visual is what TF's membrane looks like. And so I kind of put in here, you can see all of the little glyco sugars. The sialic acid here is capable of being transported into the cell. The other secret, though, about this bacteria is it is unable to make its own cell wall. So I was asking you earlier to remember the peptidoglycan and those mRNAs and GLICNAcs. It has genetically, maybe to gain energy, lost the ability to make mRNAc. Without mRNAc, it cannot make its cell wall. So it is a weakness. It's kryptonite. And so cue the Black Queen hypothesis. Again, a real hypothesis that by deleting some genes, it's gained an advantage. So TF is forced to go out now into the environment, scavenge like the dog under the Thanksgiving table, and hunt for the parts and pieces of other cells recycling. As other cells recycle their wall, like FN, then TF is able to, as a partner, grab parts and pieces of that recycling process. So it can grab pieces of mRNAc, it can grab whole sections of the peptidoglycan, and it can grab little parts and pieces. Last one I'm thinking is Fusobacterium nucleatum. So this kiddo has received a ton of limelight right now, especially with colon cancer and breast cancer, etc. So for those of you that want to know what this guy looks like, here it is. This is the facilitator. This is the bridging species. It connects the middle orange species in that pyramid to the red complex. Again, the maybe one of the most powerful relationships in the whole biofilm together is that TF Fusobacterium, connecting the orange to the red. And you can see how we've got off to the side PG connecting to FN, or early species, Streptococcus. Who knows? It's just they're connecting things to things. And I always think of it like the logs, when woods, trees are chopped down and they're sending them down the river, and you get this kind of backlog of logs. Same thing here off to the side. You can see the buildup of FN together accumulating in this biofilm. So it is used alongside other bacteria like TF. In cultures, I always think of the movie Seabiscuit, the moment when biscuit was used to help other horses win. Well, that's kind of Fusobacterium at its best. And then it's also colony-wise, it's got this kind of like fried egg shaped in the petri dish. Thought that was kind of cool. So gram-negative, it's a fusiform, so you can see those long rods. Remember, it releases the outer membrane vesicles. This was kind of the one exception. It's got a multitude of adhesions and no appendages, but instead they use adhesions. There's tons of them. So this is like molecular duct tape for FN. It breaks down protein, it does not move, it does not reduce nitrate, and it is an obligate anaerobe. It needs that no oxygen environment unless it is surrounded by other oxygen consumers. And so FN, as an orange species, has the ability to kind of latch onto both worlds. So it can cling to pioneer early strep species, for example, and then the red complex. So FN can do again a little bit like AA, a little bit of duality scenario. So middle colonizer co-aggregates with early and late. It is with us our whole lives. So FN, Heather will tell you, a lot of times it can be a commensal until it shifts into that dysbiosis mode. It may be there in decent numbers and just kind of hanging out. It's a part of the ANUG, that fusospiraketal complex, TDFN and PI, and it does release the hydrogen sulfide and methylmercaptan for bad breath. The biggest systemic environmental issues, of course, are colon cancer, cancer in general. We're talking about GI diseases, and cancer, honestly, any kind of cancer. We'll get to that. So you've got the adhesions here instead of the appendages. So the molecular glue, the way that it's sticking to bacteria, and we're going to see here the way that it sticks to tumors. Best picture I can find ever because it really highlights the tumor here, colorectal tumor. You've got these little sugars, this GalNAc, again, going back to sugars. You can see the sugar here, and Fusobacterium with these adhesions is able to latch itself onto the sugar of the tumor and right along into it. So it's capable of being a part of the metastasis conversation. And at the same time, it can actually prevent the natural killer cells from sticking. It can push them off to the side. So it's able to latch to a tumor and ward off natural killer cells. So here you see these little molecular spears and what they look like as a visual. It has the ability to survive the stomach acid, which is how it can get into the GI tract. And it just kind of highlights that importance of our stomach acid needing to be at its best and watching out for patients that are on the proton pump inhibitors, those that have, acid reflux, and it is controlled with medication and for how long, because we know who knows what's breaking through. So we've got this pathogen tumor axis that develops. So FN is now this biomarker for disease. And again, not just colon cancer, not just breast cancer, multiple cancers, because we're talking about all of those little sugars on either cells or tumors. It's able to drive cancer. It facilitates. It allows things to happen. It damages DNA through its toxins. It stimulates the tumor cell growth itself. It downregulates. You saw that natural killer cell, the way that it downregulates the immune system and can fight it off. And it leads to poorer outcomes with those that are on chemotherapy. The drama with FN. If you have ever checked out my Instagram, you will see I made a little carousel, my first one ever. Thank you, Liz, for getting me through that. The issue with FN that I have right now is there is a ton of research talking about colon cancer that's based off of this, the Nature March issue 2024, where they examined 200 patients with colorectal cancer. They found that there were these two clades. And so, actually, I'm sorry, you guys, let me back it up. Fusobacterium had a subspecies called Fusobacterium animalis that they linked to the colon cancer. And the animalis, they were very excited because they broke it down into those two clades. You can see FNA C1 and 2. FNA2, that is the one that they feel is linked to colon cancer. So they were using that to say, okay, this is the colon cancer gene portion of the clade. FNAC1 was the comparator. That was the healthy guy, and they found it in the oral microbiome. So they were using Fusobacterium animalis C1 as a comparator. The problem is, in July of 2025, These guys broke apart that research and left a scathing review. The previous article, although they said they did specific things in their study, they didn't quite always follow through and they made a couple of mistakes. And the big one was FNA, Fusobacterium animalis clade 1, does not exist. It is a totally different species. It's Fusobacterium watanabe. And the problem that they had was if you were going to compare clades to another clade that causes cancer, we need to know what we're comparing it to. And they found out we got to get that right. So they went back and totally reorganized all the Fusobacterium nucleatums, reorganized everything. They found another one, a newly defined, this Fusobacterium parent animalis, which is a commensal species. In their hunt, they found another. But the scary thing is, it's not just this FNAC2 that causes colon cancer. They found gene operons, either 1, 2, or 3 in all Fusobacterium. So what that tells me is there's a lot of research left to go, and that Fusobacterium as a whole subset has the capability of perhaps causing cancer, which I thought was really interesting. So I can't wait to see this unfold. So that's the best way I can describe that scathing review. And if we've got time, here's one more. I will try to make this quick. Prevotella intermedia. This is what we always knew as the pregnancy gingivitis oral pathogen. So we call it the hormone hound. And it too loves, an alkaline environment. It loves hormones and it loves to go after blood and the iron. So if you see Prevotella intermedia, a lot of bleeding together, don't be surprised. Especially when we've got estrogen and progesterone, it loves those hormones as growth factors, and it can substitute that for vitamin K. So instead of PG with vitamin K, it can use, hormones. In pregnancy, as our hormones surge, then it's just an overwhelming abundance of nutrients. It is a member of the orange complex. It's high up on the orange complex, though. So you will see it kind of sharing a little bit of the red complex powers, requires iron, alkaline, black pigmented colonies from the accumulation of heme and iron. PI, gram-negative rod. It does not release outer membrane vesicles. It's got a capsule. Earlier, we saw the photo of PG and PI having that capsule, so it means it's a little bit more able to withstand the environment, a little bit more protection. It's got fimbriae. It is absolutely saccharolytic. So now we have another new concept of a periopathogen that can use sugar. It can store sugar for later, and it is also proteolytic. So I want so badly to put things into a category because us hygienists, 1 + 1 = 2. Well, the biofilm does not work like that. And so there are always exceptions. And it is an obligate anaerobe, does not reduce nitrate, it does not move. Here we go, preterm birth, low birth weight. You've got your typical, you've got neurological diseases. So we're talking about Parkinson's, we're talking about Alzheimer's, cardiovascular disease. Highlighted, it's that fusospiraketo complex with ANUG. You can see the infections that it causes. It's on the tongue, head and neck. Again, not just TF's not the only one in the airway, and it loves to be below the tissue. Beautiful pictures of all of its adhesion. You can see off to the side these little guys. PI is right here in the cell. So, you've got these ideas you tell your patients, look, these pathogens climb into your cell. No wonder PG can go to the heart. No wonder AA can hide. No wonder PI is causing so much bleeding because they just nuke their way into the tissue. They have that skeleton key, that way to get into cells and hide. Interesting, again, more similarities. We had gingipanes first, and then we had dentapanes with TD. Well, now we've got an interpane, and again, another cysteine protease. So these enzymes that drive this process. Again, degrading tissue for the iron. It's going to break apart our antibodies. It loves to go after collagen. And down below, you have these little relationships, TD and PI, capturing the complement system and invading. We've got AA and PI together. Again, AA, the nice neighbor. Genes are up-regulated together, so they kind of boost each other and give each other like a, hey, pat on your back, good job. And then you have PG, PI, and FN working together to increase the risk of Alzheimer's disease. And what does that mean? Here is another fantastic visual. Here we've got gingipanes with PG, interpanes with PI, and the FAD, that adhesion with Fusobacterium, how they promote the pro-inflammatory molecules, and they start to degrade those tau proteins and increase the risk for um, Alzheimer's disease. So it's not just PG. We have these polymicrobial relationships working together to nuke the brain. And then one last photo is you have the way in which PI and PG, this is heme extraction. This is them hunting for blood together. And so you can see how they have figured out ways to work together to finally extract that iron from those red blood cells. So you will never look at a patient's bleeding the same. You will think of that little roasting fire, and you will think of them wanting to share stories and have s'mores, and they're just trying to go after all of that iron. So we're at the end of the presentation, and I want to show you this photo because this is really how extensive and expansive the oral microbiome can be to the rest of the body. And if anyone tells you you do not belong at the table or you don't have anything to say, then that is not your table, because we are here and the message is getting out and we are important and we want to be a part of medicine. And I know that medicine wants to be a part of us because we have all that beautiful saliva, all that beautiful spit as the biomarker. And just to share that this is really what we try to do at Saliva Sleuths. Heather, what a fantastic community you have built. I am honored to be a part of it. And I will say this is me and my information. Thank you guys for attending. I don't even know what time it is. Thank you for sticking around. I just appreciate the opportunity to share, hopefully, a little bit of insight into the microbial world and how the disease process has really changed for me from that brushing flossing to full-blown polymicrobial synergy and dysbiosis model. So thank you guys. Thank you. That's it.
Awesome. Thank you so much. This was like a super joy for me because it's definitely my language. I can't have, yeah, our friendship has been amazing. I appreciate you speaking for us and sharing. The super cool part is on June 24th, we are going to be diving into phase contrast microscopy. So all those fun little pictures, the spire keeps them moving. And then I think Kristen King is actually going to talk about amoebas, which are a completely different realm of the oral ecosystem with having protozoa in the picture as well, and how that impacts health as well. So if you are interested and you want to just reach out, make sure you are on my list now. I'll make sure that I send that out. But the follow-up for this at the end of the month will be phase contrast microscopy. We'll be taking a deeper dive and looking at how they move, shake, and what they look like under the microscope. So so thankful to have you here.
Does anyone more swimmy guys?
Yes, you will be looking at lots of swimmy guys. It's funny because I used phase contrast microscopy for such a long time. And in that world, they're always talking about swimmy bacteria. And there's only one that we talked about. So the red complex species, yes, the swimmy guys are bad. Spirochetes are terrible to have in there. And bristle, I think I've seen up to 9 maybe in the raw results section. But it's not just about mobility. AA, PG are not mobile. They rely on they rely on treponema maybe to pull them on in, on the outside and pulling them in. So definitely this was fantastic. Thank you so much for sharing all of the research that you've done over the many years. And it's a pleasure having you. I look forward to more.
Yeah, thank you. Thank you guys. Thanks for it's 9:46. Holy cow. Thank you for hanging in there.
And does
Woo!
anyone have any questions um, at all? You feel free to unmute and ask the questions if you have questions. I made a note just as my reminder. One, I wanted to know if you know what PG reduces nitrite down to.
Yeah, ammonia.
Ammonia. Okay.
Yeah, which is crazy. So then as it's reducing down, nitrite to ammonia, it gains that alkalinity.
Okay.
So there's a whole pathway for it. So I had to take out that slide because I'm like, oh, it's so
Yeah.
hard. Oh,
And
it's so
then,
hard
and
because
then
it's like,
I wanted to point out if PG does thrive with arginine, we have to think about the vascular system and why that makes PG such a nice home because arginine is in the vascular system. The NOS pathway for nitric oxide utilizes arginine in the vascular system. So it's a nice home with food sitting in the vascular tissues, which is why PG is so connected to heart health.
yeah, big time. I want to learn more about arginine too, like in dental products, just like a side note.
Yeah. We'll talk about that in maybe an open microbe night.
Mm-hmm.
Yeah, for
I
sure.
love
Yes,
that.
because that's something that we and Guru Nanda have put in some of ours. So I would love to know a little bit more about that because we know that arginine is like the precursor to nitric oxide.
It is,
So
but it does
in a
that's
way,
in the NOS pathway. Potassium nitrate is actually so like the Guru Nanda
awesome.
product has potassium nitrate. Potassium nitrate is the precursor or potassium nitrate is what can be utilized by the oral pathogens to reduce nitrate into nitric oxide. The NOS pathway is the arginine pathway. So that's usually utilized in endothelial cells. So we really don't necessarily need arginine for nitric oxide. However, arginine changes the pH balance and it also supports the streptococcus beneficial species. So you're increasing pH through arginine and it also benefits the beneficial streptococcus species, which reduces some of the sensitivity for arginine insensitivity because you're upping the pH level in the oral ecosystem, which changes sensitivity. Does that make sense?
Mm-hmm. Yep, there we go.
Absolutely. I I do have another question though, because I want to clarify this because when I teach in my presentations about perio is, and I just looked it up, there are studies after studies saying that perio salivary testing will be lower in perio than healthy. So I have seen there's one study that says that. So are you talking about actually in the pocket? Or are you talking about salivary testing, like just pH testing in general?
So is the question the where the pH will be?
Yeah, you mentioned that the pH is always more alkaline in the
Yeah,
pathogenic
yes,
periodontal bacteria.
yes.
And
And
from
Liz,
what I'm finding,
so Liz and I
no, go ahead.
actually, this was a good question because Liz and I, we had this conversation before we presented for the IDM that one night, and it was trying to figure out if periopathogens liked what kind of pH they liked. And it seemed weird to think that they would like a higher alkaline environment when my brain goes to someone with dry mouth and knowing that a dry environment is going to be more acidic. And I would think that a dry mouth is going to be more susceptible to bleeding, and it is. But within the pocket itself, within the sulcus, it is a different microbiome. It's a different ecology. It's a different. There's different
Absolutely.
saliva, crevicular fluid, different foods. So just like if you eat something the above everything above the gum line is getting access to sugar and oxygen, so they're burning off, like Cheetos all day long. Whereas the perio pockets, it's going to be more alkaline, it's going to be anaerobic. So it was weird. It flipped my brain to think
Yeah,
perio
get it.
guys love alkalinity because all I want to
So we've
do
got
is
it.
alkaline.
So we have to really look at that in two different ways, because if we're actually testing salivary pH, studies are showing people have a higher acidity, but
think
in a perio pocket, it may be more alkaline.
Yeah.
yes.
And
So-
I have a research article that shows like a more alkaline environment, and then after perio treatment, it comes back up to more alkaline. So yes, there's a difference between the pocket and the
right.
ecosystem within the biofilm and that pH level, and the overall oral pH within
Right.
the saliva. Does that clear?
That makes complete sense, because the pocket is a totally different environment, a totally different region. It's protective, and it's going to help. We don't have a ton of also cavity pathogens. We don't have a lot of that definitely in the pocket as much as periodontal. We have more of those on the surface top where there's more acidity. So, they're thriving where they can thrive, and they know that that's where to go. I always tell my patients that your bacteria is so smart.
It's like,
they know exactly where they're going to go.
There was another study too where they were using ascorbic acid, like vitamin C, into the subgingival pocket because it was acidic as a treatment for perio. So I had heard that too. So the thought of putting acid down in the pocket to try to change their environment. And you think about even,
When you're worried about dentin, though, I mean, the problem is we're dealing with the whole complexity of the oral cavity is that you're dealing with two completely different structures. You're dealing with gingiva and gingival tissue, and sustainability of that. And then you're talking about, well, actually 3, bone. And then you're talking about tooth structure. And so they're not always conducive to each thing. I mean, we have to be careful, because one treatment may actually impede or hurt or be more problematic in another area. But there you go. That's why we have you.
The butterfly effect.
But and you know what, isn't that the most important key, that any product you put in someone's hands, that it has to be chosen with purpose, that you don't want the patient just going down the Listerine aisle and picking out whatever. We really want to we're the experts. We know what they need and they don't. And so it's up to us to kind of know what best is going to suit their needs at that moment. And even in the microbiome, we talk about like the strep and then AA together. You've got 2 totally different pHs designed for survival. And there will be more when we break down the chromogenic stuff, Heather, because it gets nuts. There's going to be a whole episode on us breaking down cavity bacteria and weird stuff they're doing. It's the battle. It's the battle.
Yeah. Holly has a really cool article coming out soon that she's working on, where she took the sticky yellow plaque that you get on a crown margin. We all have plucked and scraped that like film off underneath, and she sent it into Bristol, and she had multiple samples. So now she's analyzing. And I can't wait for that article to come out. I have put that under a microscope and just saw cocci.
Are we going to find a lot of yeast also? Can we find that? No, not at all.
Yeah. Not saying not necessarily. And I'll tell you what, I have had the great honor of reaching out to Dr. Tanner and emailing her. She is alive still. And we've written emails. It's the best email. Ugh, it's like just to think that she's alive and wrote back. And I shared with her, my questions about this, and she wrote back, and her first thought was yeast. Yeah. Which was really interesting. That's not what's on the reports. But then Heather, I would need you at some point to kind of confirm, Bristol's testing candidas.
Yeah. We have 4 different species of candida. But who knows if there's others that we just don't, we're not. I had them put Candida in the carry score. It doesn't push the score at all. It doesn't push the algorithm. But in my findings, clinically using phase contrast microscopy, Candida was a major issue for a lot of gum line cavity issues that I was seeing. That's why I started bristle testing, because I didn't want to do multiple tests with oral DNA to get the answer there. But there's another course that I ran into, and he talks about the Candida conversation. But Candida is absolutely a pusher. I think also with the biofilm, it's synergistic with strep mutans and makes it really sticky and helps with the acidic component of a biofilm, but then also it's inflammatory. So it's going to feed, certain pathology, but also when swallowed, we have the gut impact and CFO, and then we have cytokines and interleukins in more of the inflammatory response that gets hyper exasperated. So I think Candida is a real problem for some individuals. And honestly, even the amount of Candida we find in the oral microbiome doesn't always indicate what that level might be in the gut. So I've had some lower levels in salivary testing, had people go for gut testing and their Candida levels were really high in the gut. And then when we have that gut component, we have cytokines, interleukins, tumor necrosis factor alpha that feeds into the perio story.
That's awesome.
You guys are amazing.
Everyone here is amazing.
Yeah.
Everyone is like so brilliant, and everyone has their own special niche.
Now, Holly, you are the next Tanner. You're going to get a Nobel Peace Prize.
Yes,
Right. I don't know about that.
At least for your slides. I can't. Did you make all those slides? Because I'm like just so impressed of your little.
Yeah.
I see. I don't do like adult color books, but Heather and I, we joke about how we like to make slides.
Yeah.
Well, you're talented.
Oh, thank you. I need it for visuals. It takes me a long ass time to try to figure out what glycoproteins and sugars on cells, and it's like, oh my God, like I just could not. And then all of a sudden it clicked. And, Dr. Bertozzi in that YouTube video, and she just laid it all out and made it so easy to understand. I was like, okay, now it fits. Now I get it.
Yeah, beautiful job. Presentation was awesome.
Oh, thanks, guys. That's fun. Guys, thank you so much. This kind of keeps me feeling like, you're all my people. Makes me feel good.
Does anyone else have questions? Patty? We have a Patty. Patty, I've not met you. I don't think. Are you there? No. All right.
Past their bedtime.
Probably, yes. We will wrap then. Please join Saliva Sleuths if you like conversations like this. This is a little bit of a hint of what our conversations together on the open microbe nights are. It's oftentimes Holly and I talking,
nerding out,
really nerdy insight of the oral microbiome and how everything works, and then even products and things that we kind of talk about on selection of what we're doing with those products. Thank you for mentioning desiccation therapy, laser bacterial reduction, because when we have embedded tissues, we have to do more than just hand instrumentation and cavitation. So super. All right.
And I have a question. I do have a question just as a newbie, because you both know that this is not I am like just your basic hygienist, and I want to take and just give it in the normal terms. But we do not want to get rid of everything. That's the key. We need red and orange. We need them, but only in a controlled. We don't want overpopulation. Am I right? Is that what we're talking about? Because we can't eliminate all red.
We can't eliminate all red. I don't like seeing PG or AA in the picture. I don't see them very often. Treponema denticola I see, from time to time. Tannerella, there very often, especially with Fusobacterium. So you can start to see that escalate. I do have a little bit more of a non-tolerance for PG and AA, and those ones, Treponema. I think the bristle test, like I said, I think I've seen about maybe 9 species of different Treponema, and I don't really have a lot of tolerance for those ones either because of the pro-inflammatory impacts and the systemic impacts of Treponema species in general. So when I do see AA or PG or those red complex species, I'm usually encouraging offices to utilize lasers. Desiccation therapy is a wonderful tool to help with that. And that's also going to help with the tissue healing as well through that process. So I'm not a huge proponent for antimicrobial use like amoxicillin and metronidazole. We could maybe, I was just mentioning this to one of my other sleuths on a result review, like we have things like maybe oregano oil tablets that we could take. Oregano is a very broad spectrum antimicrobial, not something we should be taking. We should think about using that as an antibiotic type of regimen. Or there's ozone capsules that you can utilize. Ozone is O3. We're dealing with anaerobic bacteria. And it can help stimulate the mitochondria and assist the immune system as well. So maybe thinking outside the box of antibiotics, just because antibiotics, when we use them, these bacteria have genetic capability of becoming antibiotic resistant, and we turn those genes on when we start using products like that. Even products like chlorhexidine up the susceptibility of the genetic capability of becoming antibiotic resistant. So we have to be really careful about antibiotics.
Heather, what does that make if you're resistant? What does that lead into?
It's the resistome. The butterfly effect. In the Saliva Sleuths, we have an open microbe night. Check out the butterfly effect conversation. We're hinting to that one. So perfect. All right. Thank you so much.
Thank you, guys. Thank you. Until we meet again.